Hi, Michelle Glass here, and this video is to help with Lab 12, looking specifically at this section on DNA fingerprinting. Now, DNA fingerprinting can also be described, or DNA fingerprint, I should say, can also be described by this fancy way. You can call it a restriction, fragment, length, polymorphism, or RFLP for short. Now, that maybe doesn't make a lot of sense now, but hopefully as we get to the end of our video, you'll be able to understand that and sound just as fancy. Okay, so to start, let me go back here. This is where we should be. When we're talking about DNA fingerprinting, there's going to be three major steps. And the first major step is to actually isolate that DNA from your sample. And in order to isolate the DNA from the sample, you have to do three steps. And this is all actually pretty easy. This is described as a kitchen lab. So this is something you could do in your kitchen at home. So the first step is to homogenize or release the DNA from the cell. And you can do this mechanically with a blender. So in our lab, sometimes we'll take frozen strawberries, throw them in a blender, turn it on, and we're not making daiquiris. Instead, we're trying to break up the cell wall, break up the cell membranes, break up the nuclear envelope, and release that DNA into solution. When you do that, that DNA then is mixed with all the components of the cell, and you're going to see a lot of proteins that are going to be attaching to that DNA. So this second step here is described as deproteinization, which means remove proteins from that DNA. And you can do this with one of two ingredients that you might have in your kitchen. So you can use like a Dawn dish stove, or you can use a meat tenderizer. And either one of those will do the trick. And then the third thing that you do, and this you might not have in your kitchen, is precipitation. You can do this with either ice cold ethanol, which is what we do in our lab, or maybe you have rubbing alcohol, isopropyl alcohol at home, so you could do it with that. And what you would do is you would mix together the ice cold ethanol with that homogenized, deproteinized mixture that you took out of the blender. You added the meat tenderizer. You can see my diagram there below. What you're going to see is a separation of all of the cell stuff with the DNA in that test tube, and you can actually swirl up that DNA on a glass rod if you're super lucky. So I rarely have had that kind of success, but I have, of course, seen that DNA separated out, and it's a little disappointing we can't do it this semester, but to be honest, it just looks a little bit like a snotty smear, so it's really not that exciting. Okay, once you have that DNA isolated, you need to do your second step, which is described as, polymerase chain reaction, or PCR. Now let's go back for a minute and think about our DNA isolation. If we take a whole bunch of strawberries and throw them in a blender, then we're going to get a whole lot of DNA out of that sample. If we're doing this as like a crime scene analysis, you may be lucky, hitting all the wrong buttons. You might be lucky just to get a small, tiny sample of DNA, like 1 or 2 cells. In that case, you really don't have enough DNA to use. So that's the purpose of polymerase chain reaction, or PCR. PCR is a technique that allows us to amplify our DNA, meaning make more from a really small sample. And this is really what's made all of the DNA analysis that we can do possible. This is done with a machine. It's super easy. You're going to add in your DNA sample that you've isolated. You're going to add in the special primers that you need. This primers select a specific sequence of DNA to copy. So we're not actually copying the entire chromosome, not the entire DNA from the cell, just a small section that we're interested in. You need to add, of course, your ingredients to make new DNA. So that's going to be the monomer, the nucleotide. And you're going to need some enzymes that are going to be involved in replicating DNA, such as DNA polymerase and helicase and so forth. when you put them in a machine and you program it, and the machine takes you through cycles of hot and cold and allows for this PCR process to occur, and you can set it in and you can go home and eat some pizza and watch a movie and go to sleep and wake up the next day and go back to the lab, and you have a whole lot of DNA from that original teeny tiny sample. So if you look at the picture, when we talk about amplification of DNA, we can start with one strand and it copies to give you 2, and then both of those strands can copy to give you 4, and then 4 strands can copy until you get 16, and so you get exponential replication. And so this allows you to have going from a very tiny sample to like a whole lot of DNA, and now you have a usable amount. So we started out with DNA isolation. We move on to polymerase chain reaction, which allows us then to go to our third step, which is described as gel electrophoresis. So this is how we actually get to see our results. Now, before we actually stick our DNA into the gel and perform gel electrophoresis, we do need to treat our DNA with what's described as a restriction enzyme. A restriction enzyme is actually going to be like a tiny pair of scissors that's going to go in and cut your DNA, but it's not just going to randomly cut here, here, and here. It's going to cut at a specific location. So what we see is that these restriction enzymes are enzymes, so they're highly specific. They go along your DNA strand and they can read a certain sequence, and after that sequence, they'll cut every time. So I've given you an example here of a restriction enzyme described as EcoR1, and that tells us that it was derived from an organism, Escherichia coli, E. coli, and R1 stands for the first restriction enzyme that was isolated from this organism. So restriction enzymes do exist in nature. These are typically found in microbes, and it's going to be important in a lot of those microbes' functions, okay? And we're just able to use these. And so what we're going to do here is cut our DNA into fragments. Now again, each restriction enzyme has a specific region it recognizes and makes a specific cut. So to really enhance your analysis, to make it more robust, more reliable, you want to add more restriction enzymes. So the more restriction enzymes you use in your analysis, the more robust, the more reliable your analysis is going to be. All right, so how do we actually then do gel electrophoresis? We're going to, I'll show you all this in a diagram, but we're going to add DNA to an agarose gel. Agarose is derived from seaweed, and it basically acts like a gelatin for us. So it's going to make something that's going to be kind of like a jello. And we're going to add some electricity, and that's going to let us separate these cut fragments out based on size. So that's essentially what we're going to do. Let's look at it in a little bit more detail. Okay, so this is typically what an agarose gel is going to look like. It's usually kind of rectangular in shape. They're usually, fairly thick. And again, it's going to be like a little slab of jello, gelatin, but in this case, it's composed of agarose. And when you make these gels, you're going to create these little wells or indentations at the top of the gel. And it's inside those indentations that we can actually load our DNA. Now remember, this DNA has been cut with restriction enzymes already. And we're going to use in this example 3 different samples. So we're going to load sample A, sample B, and sample C into our gel. The next thing that we do is turn on electricity. And when we do that, we're going to create a negative end and a positive end. Because our DNA molecule is a negatively charged molecule, it will be attracted to that positive end. So that DNA is actually going to be moving through the inside of the gel, being pulled based on this attraction to this positive end. So imagine if you're swimming through syrup, right? It's going to be a lot harder to swim through the syrup than it would be to swim through water. And think about different sizes of fragments or different sizes of people. So a smaller body is going to move through that syrup a little bit easier than a really big body. So a smaller fragment is going to move through that gel a little bit faster than a thick fragment. And so that's going to be what allows us to separate these fragments based on size. Typically, we let our gel run for about 45 minutes. This can vary. If you let it run for too long, all that DNA will just run completely off the gel and you won't have any data. If you don't let it run for long enough, then those fragments won't migrate or move the length that they should, and you may not be able to see all of those fragments the way that you should. Again, remember those big fragments are moving more slowly, so they're going to stay closer to those wells, and those smaller fragments are moving much faster, so they're going to make it more towards the bottom of that gel. And notice then that we're seeing a different combination or pattern of fragments for our sample A, our sample B, and our sample C. So what we see is in each case that restriction enzyme cut 3 fragments, but we have different sizes. And we can tell they're different sizes based on where they are located on the gel. In order to match a DNA fingerprint, what you look for is a matching pattern of those fragments. So if we look at this, we can see that my sample A and my sample C have 3 fragments that are placed at the same locations on the gel, so that's considered a match. When we look at sample B, it also has 3 fragments, but they're at different locations, and so it's not a match. So if we were to say that sample A is crime scene evidence DNA and samples B and C are suspects, then we would say suspect C is a match. Now this analysis is not super robust because I'm only using one restriction enzyme, so there's room for error, okay? By using more than one restriction enzyme, I can enhance my analysis. In that case, then you're going to have more than one lane of information for A, more than one lane of information for B, and more than one lane of information for C. You would want to make sure you're matching up apples to apples and oranges to oranges. What I mean is if you have restriction enzyme 1 and restriction enzyme 2, you want to make sure that you're comparing all of the lanes or all the DNA cut with restriction enzyme 1 together, and then all of the DNA cut with restriction enzyme 2 together. Lastly, then, let's go back and see if we can better interpret our fancy name for a DNA fingerprint, RFLP, restriction fragment length polymorphism. So restriction fragment is just talking about that little section of DNA, that little cut that we get by using a restriction enzyme. Length polymorphism means that the length is going to vary. So polymorphism is multiple shapes. So when we interpret RFLP, it's basically just a fancy way of talking about the pattern of fragments that we get through the process of gel electrophoresis. And that's it.